Dear student, today we are going to learn 8th topic of science 2, and that is cell biology and biotechnology. All of you know cell is the structural and functional unit of living organism. A group of cell performing similar function, having common origin and having same structure is called tissue. Different tissues perform different functions in the body. The study of cell structure, cell organelle, cell division, and other aspects of the cell is called cytology. Last year, we have learned how the new plant is produced by the technique of tissue culture. Now, we are going to learn about stem cells. These are the special types of cells present in the body of multicellular organism, which can give rise all other types of cells present in the multicellular organism. In this topic, we are going to learn stem cells, particularly in human being. All of you know the male and female gamete unite and form a diploid zygote from which new living organism formed. These zygotes start dividing mitotically and form a mass of cells in which all the cells are same. These similar cells are called stem cells. On further dividing, these cells form different types of tissues which performs different functions in the body. This is called differentiation of stem cells. That means stem cells on differentiation form different types of tissues which perform different function in the body. However, some stem cells are present in some parts of the body for long duration. The different locations in which these stem cells are present are umbilical cord, by which a fetus is joined to the uterus of mother. Stem cells are also present in the blastocyst, which is formed in the early stage of embryonic development. Beside this, the stem cells are also present in the red bone marrow and adipose tissue of adult human being. These stem cells present in the different location can be produce different types of tissues and the degenerated part of any organ. Depending on the source, the stem cells are divided into two types, and they are embryonic stem cell and adult stem cells. The embryonic cells before differentiation are called embryonic stem cell. Nearly 220 different types of cell in human body are formed from this single type of cell, that is embryonic stem cell. It has been found that if these stem cells are collected before beginning their differentiation, that is on 14th day formation of zygote, and preserved in the laboratory by keeping them in liquid nitrogen at temperature minus 135 degrees Celsius to minus 190 degrees Celsius, then as per the stimulus they can be transformed into desired type of cells and therefore desired type of tissues, and finally we can convert them into a desired organ which we require. The second type of stem cell is adult stem cell. These cells are obtained from the adult body either from the bone marrow or adipose tissue or blood. These stem cells can be used for regenerative therapy and organ transplantation. In the regenerative therapy, the stem cells are used to replace the dead cell. In case of condition like diabetics, myocardial infarction, Alzheimer, Parkinson disease, and so on. In case of organ transplantation, stem cells are used to replace the organs like kidney, liver. We can produce this organ with the help of stem cell and can be transplanted. Now let us learn in detail organ transplantation. अनेक कारणामुळे जसे ॲक्सिडंट, इन्फेक्शन, आजार किंवा वयोमानामुळे वेगवेगळे ऑर्गन्स हे कमी इफिशियंटली काम करतात किंवा ते त्यांचं काम पूर्णपणे थांबवतात. त्यामुळे अशा व्यक्तींच्या जीवाला धोका निर्माण होतो. परंतु या व्यक्तींना जर हवे असलेले ऑर्गन मिळाले तर त्यांचा जीव वाचू शकतो. परंतु यासाठी कुणीतरी ऑर्गन देणारा दाता असावा लागतो. उदाहरणार्थ प्रत्येक माणसाच्या शरीरात 2 किडनी असतात, ज्या ब्लड फिल्टर करून युरिन तयार करत असतात. हे सर्व काम एक किडनी सुद्धा करू शकते आणि त्यामुळे अशा व्यक्तीची दुसरी किडनी ही डोनेट करता येऊ शकते. परंतु दुसरे ऑर्गन्स जसे लिव्हर, किडनी, हार्ट, आईज हे मात्र दात्याचा मृत्यू झाल्यानंतरच घेता येऊ शकतात, ज्याला आपण आफ्टर डेथ डोनेशन असं म्हणूया. ऑर्गन डोनेशन होत असताना ब्लड ग्रुप, वेगवेगळे आजार यांचाही विचार करावा लागतो आणि मगच ट्रान्सप्लांटेशन शक्य होऊ शकतं. देहदान आणि अवयवदान ही काळाची गरज आहे, कारण मृत्यूनंतरही आपल्या शरीरातले ऑर्गन्स हे काही वेळापर्यंत जिवंत असून ते वापरले जाऊ शकतात व त्याचा उपयोग जगण्यासाठी होऊ शकतो. दॅट मीन्स वी कॅन सेव्ह द लाईफ ऑफ अदर पीपल आफ्टर द डेथ. दॅट मीन्स लाईफ ऑफ मेनी पीपल्स कॅन बी सेव्ड बाय द ऑर्गन अँड बॉडी डोनेशन. त्यामुळेच बॉडी डोनेशन म्हणजेच देहदान हे उत्कृष्ट दान आहे. नाव लेट अस लर्न अबाउट बायोटेक्नॉलॉजी. Biotechnology means bringing out artificial genetic changes and hybridization in living organism for human welfare. Various branches like genetic engineering, molecular biology, cytology, biochemistry are included in biotechnology. आणि या बायोटेक्नॉलॉजीमुळे ॲग्रिकल्चर आणि फार्मसी या क्षेत्रामध्ये प्रचंड प्रगती झाली आहे. भरघोस उत्पन्न देणाऱ्या पिकांच्या जाती त्यामुळे अस्तित्वात आल्या. फार्मसीमध्ये वेगवेगळे वॅक्सिन्स, अँटीबायोटिक्स, व्हिटॅमिन्स, हार्मोन्स तयार करता येऊ लागले. तसंच ॲग्रिकल्चरमध्ये वेगवेगळ्या डिसीज रेझिस्टन्स स्पेसीस, स्ट्रेस रेझिस्टन्स स्पेसीस तयार करता येतात. त्यामुळे ते कुठल्याही रोगाला बळी पडत नाहीत. हवामानातील बदलही ते सहज पचवू शकतात. आणि ह्या बायोटेक्नॉलॉजी, टिश्यू कल्चर या टेक्निकचा वापर करून जेनेटिकली मॉडिफाईड क्रॉप, ज्याला आपण GM क्रॉप म्हणूया, जे भरघोस उत्पन्न देतात, किडीला सहसा रेजिस्ट असतात किंवा टेम्परेचर किंवा इतर व्हेरिएशनसाठी त्या रेजिस्टन्स असतात, अशा व्हरायटीज जशा बीटी कॉटन, गोल्डन राईस, हर्बिसाइड टॉलरंट प्लांट तयार झाले आहेत. आजच्या काळात आपण ज्या काही भाज्या, फळं, कडधान्य, अन्नधान्य हे खातो हे संकरित आहेत, हायब्रिड आहेत. इन ॲनिमल हजबंड्री also, many hybrid varieties of cows and buffaloes are developed to improve the quality and quantity of milk production. For more amount of milk, meat, wool is available now. In human health, early diagnosis of disease is possible, which will help to early treatment of the disease so they can get cured. Hormone insulin is used in the treatment of diabetic patient. Earlier, it was obtained from the pancreas of horse, but nowadays this insulin is prepared in the laboratory by using biotechnology from the bacteria. For this purpose, the human insulin gene is introduced in the genome of bacteria. Various vaccines and antibiotics are also made by using the same way. Due to vaccine, we can acquire permanent or temporary immunity against the particular disease. Earlier, vaccine were made by killing or half killing the pathogen and then introducing into the body of a organism. Nowadays, the vaccine is prepared by using antigen, which is prepared in the laboratory with the help of gene, which is isolated from the pathogen and used as a vaccine. Such vaccines are safer to use. Now, let us learn about the term edible vaccine, whose work now is in progress. Potatoes are being produced with the help of biotechnology, which are called transgenic potato. This potato will act against the bacteria like Vibrio cholerae, E. coli. Consumption of such transgenic potato gives immunity against the disease like cholera and the diseases which are caused due to E. coli. Now, let us learn about interferon. This is a group of small size protein molecule which are used in the treatment of viral diseases. These are produced in blood. Nowadays, the transgenic E. coli are used for the production of interferon with the help of biotechnology. Now, let us learn about gene therapy. This gene therapy is used to treat the genetic disorder in somatic cell. For example, phenylketonuria can be treated by gene therapy. Now, let us learn what is meant by cloning. Production of replica of any cell or organ or the entire organism is called cloning. जे एखाद्या वनस्पती किंवा प्राण्यासारखा हुबेहूब प्राणी तयार करता येऊ शकतो. In this technique, there is no need of sperm to produce new organism. A clone can be produced by fusion of a nucleus of somatic cell with the enucleated ovum of anybody. A ship, Dolly, was born in Scotland by cloning technique on 5th July 1996. In therapeutic cloning, the stem cell can be derived from the cell formed in the laboratory by the union of somatic cell nucleus with the enucleated egg cell. Hence, various diseases can be treated with the help of this stem cell. Similarly, various industrial products, which is called white biotechnology, can also be produced through less expensive process. For example, alcohol production from sugar molasses with the help of transgenic yeast. Now, let us learn environment and biotechnology. Various environmental problems can be solved with the help of biotechnology. For example, sewage is rich organic matter. If it is released in the water body, it causes pollution, reduces the level of dissolved oxygen, and adversely affect the aquatic life. So, as a remedy, this sewage should be released into the river water only after oxidation with the help of microbial technique. Microbes are also useful on the large scale production of compost. Bioremediation, biopesticides, biofertilizer, biosensors, etc. are some of the new concepts in biotechnological method. Bioremediation means absorption or destruction of toxic chemicals and harmful pollutants with the help of plants and microorganisms. If the plants are used for this purpose, then it is called phytoremediation. Grasses like alfalfa, clover, and rays are used in photoremediation. Pseudomonas bacteria are used for changing the hydrocarbons and oil pollutants from the soil and water. The fern Pteris vittata can absorb the arsenic from the soil. Genetically modified variety of Indian mustard can absorb selenium from the soil. Sunflower can absorb uranium and arsenic. Now let us learn about food biotechnology. Food items like bread, cheese, wine, beer, yogurt, vinegar are produced with the help of microorganisms. These food items are probably the oldest one which are produced with the help of biotechnology. Now let us learn about DNA fingerprinting. DNA sequence of each person is unique, like that of fingerprints. म्हणजेच प्रत्येक व्यक्तीचे फिंगरप्रिंट्स आणि DNA सिक्वेन्स ही स्पेसिफिक असते. त्यामुळे आपण एखाद्या व्यक्तीची आयडेंटिटी ओळखू शकतो. This is called DNA fingerprinting and it is mainly used in forensic science. It is used to identify the criminal. It is also used to identify the father of any child. Now, let us learn the important stages in agricultural development. The first stage is green revolution. We are self sufficient in food grain production due to development in the agriculture and by application of biotechnology. We are self sufficient in food grain production, which is called green revolution. Dr. Norman Borlaug is called father of Green Revolution, as well as Dr. M.S. Swaminathan also have valuable contribution in Green Revolution. White Revolution. Self-sufficiency in milk production is called White Revolution. Dr. Verghese Kurian is the father of White Revolution. Blue Revolution. The production of various useful aquatic organisms with the help of water is called blue revolution. Fishes are cultivated in farm ponds. Biotechnology also plays the important role in the production of insecticide or for organic farming. In the agriculture, we can cultivate many medicinal plant as well as the fruit can be processed and different types of jams jellies are obtained from there.